Well, good morning. Delighted to see you all here for a very special occasion on the UT Southwestern campus when we have the opportunity to welcome and hear from Dr. Francis Collins, the current director of the National Institutes of Health. I think everybody in this room will appreciate that the NIH has been an engine and catalyst for progress in biomedical sciences and the resulting benefits to the health of our country and the world for decades now. And certainly it is a cornerstone of what makes our faculty, working with our students and staff, be able to fulfill our commitment to improve health through research. Dr. Collins, as a physician scientist, brings exceptional accomplishment and experience to his role. His turn towards medicine followed completion of a doctorate at Yale in physical chemistry, when a course in biochemistry directed him in a different direction than he started. He completed medical school at the University of North Carolina, Chapel Hill, and then joined the faculty thereafter at University of Michigan, establishing a record of seminal contributions as a geneticist. The process of chromosome jumping that he developed as a technology that advanced the ability to identify disease-related genes soon resulted in the identification of the underlying basis of many genetic disorders, most especially as a milestone, the identification of CFTR as the gene underlying the disease of cystic fibrosis. He was called upon to come and lead the efforts at the NIH in 1993, and was appointed eventually to be the director of what is now the National Human Genome Research Institute. It was under that aegis that he led the Human Genome Initiative that culminated in the announcement in 2000 of the first draft of the human genome. In 2009, President Obama nominated him for his current role, and he was approved unanimously by the Senate and has served as a distinguished leader of the National Institutes of Health, being a trusted spokesperson on Capitol Hill and a leader who has forged new directions for the NIH, including such initiatives as the NCATS, the National Centers for Advancing Translational Science, the BRAIN Initiative, the Accelerating Medicines Partnerships, and recently the Precision Medicine Initiative. So we have in Dr. Collins as the director of the NIH, a visionary as well as someone who speaks from the experience of what it takes to move science along as a practicing scientist, something he continues to this day. And so it is a great honor to have him here at UT Southwestern and to hear from him in his prepared remarks, to be followed by questions which will be fielded by our Provost for Basic Research, Dr. David Russell. Dr. Collins' topic today is exceptional opportunities in biomedical research. We welcome you to UT Southwestern.
Well, thanks for that very kind introduction. It's wonderful to be here at UT Southwestern. It's been a few years since I've had the chance to visit with you all. And this is a place of great strength where opportunities and developments in medical research are happening almost daily. And it's always fun to be able to get outside of the Beltway, especially now, and have a chance to come and see what's happening in one of our nation's great medical research institutions. And that's what you all are. So it's great to be here with you. If I can have my slide up, there we are. I'm going to try in a fairly broad way to paint a picture of some of the exceptional opportunities that I see right now as contributing to the sense that we all have that this really is a remarkable moment to be involved in biomedical research. And I won't be able to drill very deeply into any of them, so this is going to be sort of dancing across the mountaintops, but hopefully in the questions we can dig into some of those other issues as well. So just to remind you, NIH has a twofold mission, and it's well represented here. Science in pursuit of fundamental knowledge about the nature and behavior of living systems. That's our basic science effort. That's 53% of what we do, and that hasn't changed over the last couple of decades. But also the application of that to reduce the burdens of disability and illness and to extend healthy life. That is the other part of what we do. And you all who are here at this institution are engaged in both of those. And of course, there's the translational steps in between, which in many ways everybody is trying to improve the opportunity for those kinds of handoffs to happen and being quite successful at it. We fund science across the U.S. Most of our money goes outside the Bethesda headquarters to institutions like this. Currently, Texas receives over $1 billion of NIH funding, and you guys right here are doing pretty well with $175 million of that. I hope you're spending it wisely, and I believe you are. Because, of course, this is all based on the most rigorous peer review system in the world, which, while not perfect, is working pretty well, and we are always trying to come up with ways to make it work even better. And when I look to see what you all have been doing lately, I just kind of looked at your website to see your most recent announcement of advances. So this is just over the last month or so. Here's 10 examples. And of the 10 examples I found, maybe there's 8 here that were all supported by NIH. So we're proud to say that we're a contributor to the amazing work that's going on here. So I thought I'd try to divide this up into 4 quick themes. Again, trying to paint the picture here of the breadth of what medical research right now is all about, beginning with basic science research and then moving into more applications, but also I want to talk about training because that's so crucial to our future. I'm glad to see there's a lot of young scientists here because you are the people that we are most interested in making sure we're providing you with a stable trajectory so you can chase after those innovative ideas that I know are bubbling up all around us. The basic science part, maybe it won't surprise you that I do need to say a word about the way in which genomics has played a pretty central role in a lot of the discoveries that are now possible that I think are pretty amazing to contemplate in terms of what a single laboratory can do that one would not have thought possible a couple of decades ago. The success of the Genome Project, of course, led to our having publicly accessible the entire script for the Human DNA Instruction Book and that of a lot of other organisms. But we need to understand how it works in order to bring this to the bedside. We need to change this image a little bit where there's actual revelations happening by the use of genomics, and that is coming forward, and I'll say something about that in a minute in terms of precision medicine. But we had a lot of work to do, and still do, to try to provide functional information about the genome in order to have that transition happen. And you see here a whole lot of banners of projects which were funded by NIH in a way that brought many investigators together from multiple different disciplines, working to produce databases or tools and technologies and giving them away so that everybody had access to them. And I think that has been one of the empowering aspects of this. And that includes applications to specific diseases like cancer or to particular kinds of databases like the microbiome and a whole lot of other things as well. As a result of all of this capability in genomics, laboratories are transformed. My lab, which 10 years ago was primarily looking at hereditary factors in diabetes, has completely transformed into an epigenomics laboratory, trying to understand how those diabetes risk factors have an effect on gene expression in pancreatic islets and other relevant tissues. And my lab, which used to have people working at the bench, and now has a few, but most everybody else is doing computational analysis from free databases, and I'm sure that's happening here as well. One of the other consequences of this, of course, is that we have gone very rapidly from knowing that there were thousands of diseases to knowing what their molecular basis is. If you see the way in which that information has grown, we are now up to about 6,000 conditions for which we know the molecular basis. Most of these are relatively uncommon genetic disorders. The bad part of this story is that only about 500 of those currently have treatments that have been proven successful. So we have a big gap there between what we know and what we can do. But even getting a diagnosis can be awfully helpful in many circumstances, and recognizing that there are still many individuals out there for whom diagnosis has been elusive, despite many visits to doctors. We started the Undiagnosed Diseases program at NIH, and it's now spread across the country as a means of bringing together experts across many different medical disciplines and having them read hard to see if they could sort out and get an answer from those individuals who've gone through many frustrating experiences without having a clear sense of what the diagnosis is. Yeah, you can see some of the statistics here. Bill Gall, who runs this at NIH, I think has been really quite an icon of how this kind of program could go forward. And yes, we have now expanded the model to multiple other centers across the country, and many across the world are now adopting this same view. And as you can see here, we do get a diagnosis, not all the time, maybe 25, maybe 30% of the time. And some of those times, it makes a really big difference in terms of understanding what therapy would be appropriate. All of this coming about really because of the facility of being able to do imaging, but particularly to do genomics at scale. And with the cost of a genome sequence now, I think, credibly under $1,000 for a whole genome sequence, and falling to the point where one can imagine the $100 genome in the next 5 years, this becomes transformative for the practice of medicine. Another area of basic science importance with obvious applications and implications is brain. This project, which started at NIH, but in collaboration with NSF and DARPA and a couple other agencies 3 years ago, the Brain Research through Advancing Innovative Neurotechnologies, the BRAIN Initiative, is going great guns and has now acquired a budget approaching $400 million a year, thanks to the Congress and the 21st Century Cures Act. And the goal of this, which is outlined in this document, which was put together by a remarkable dream team of neuroscientists led by Corey Bargman and Bill Newsom back in 2014, is to spend the first 5 years of this 10-year effort really building technologies that allow you to see what's going on in real time in complex circuits in the brain where the action is, and which have largely been out of view because we didn't have the technologies to look at such complicated circuits. And we're getting there with the resources that are going into this. Obviously, a lot of this depending on model organisms, but ultimately we aim to figure this out and see how it is that your brain is able to carry out remarkably complex activities at the speed of thought in a way that will probably completely revolutionize our understanding of neuroscience and maybe even of how computers should operate. We can learn from Evolution's lab notebook here. We're just beginning to learn how to read those scribbles. So all of this is going to be, I think, an enormously exciting time for anybody who's looking for a pathway and trying to decide where to land in this big, broad landscape of life science. The initiatives on the brain are going to be one of the most exciting spaces. And I was thrilled to hear this morning that UT Southwestern aims to try to make a big investment in that space by building an initiative that brings together the kind of expertise that's needed here across technology development, computational skills, and basic science of neurons, and also imaging capabilities, but connects that up with clinical applications in a way that we hope can speed up what we desperately need, which is new ideas for conditions like Alzheimer's disease or Parkinson's or autism or schizophrenia, down this long list of conditions that affect the brain. So those are a few snapshots of the basic research enterprises that are attracting attention. And again, there's lots of others I'm not touching on because of time, but those are two, genomics and neuroscience. But what about the translational part? How are we doing in that? Well, I feel a little bit like the guy who brought coals to Newcastle to come to this institution and talk about translation, because after all, when I think of UT Southwestern, I think of the long tradition that has been in place here, and it basically is the cardiovascular disease success story that now results in the fact that deaths from heart attack and stroke are down by more than 70% over where they were 40 or 50 years ago. And it almost happened gradually enough that most people in the general public haven't noticed. And all of this, I'm fond to say, has been accomplished through NIH support of researchers, many of them here, that cost the average American the equivalent of 2 lattes per year. That's not bad. Somebody told me I should change that analogy and now talk about it costs you 6 donuts per year. But anyway, it's not much money. And cardiovascular disease, of course, we really didn't know much about. What were the reasons for that going back 60, 70 years? The Framingham study, which is in many ways the granddaddy of all cohort studies, and I'll come back to that also when I tell you about the new one, the All of Us program, showed that high cholesterol was a risk factor, but other things as well, hypertension. But we weren't clear why cholesterol was the risk factor. And of course, I have to actually highlight here a couple of individuals, and I'll get to another one in a minute, who made this story go from obscurity to the central understanding of what's happening in cardiovascular disease. And of course, I also have to mention that Brown and Goldstein were formerly postdocs at NIH. So any of you who are looking for a good place to do a postdoc, that's still true. And their discoveries, which led, as you know, to the Nobel Prize and to the development of statins, changed this whole understanding of cholesterol into a very highly refined science. And along the way, they recruited a few other people to work on this to see if there were some other things that could be learned about this. And I want to highlight my friend Helen Hobbs, who's been a fantastic contributor to this field and also a very capable advisor to me in her role as one of the members of the advisory committee to the director with the Dallas Heart Study and with her colleague Jonathan Cohen, discovering patients who had effectively knockouts of PCSK9 with very low levels of LDL cholesterol and very low heart attack risks, making it clear that this was a natural experiment that tells you of a perfect opportunity to develop a therapeutic to further lower cholesterol in people who are not responding effectively to statins, especially those with familial hypercholesterolemia. And as you know, that resulted in a very good outcome that is all around us right now. But in terms of this translational stuff, I think we should all look closely at what's happening right now with the development of gene editing at a scale and a precision that I would not have imagined possible a few years ago, and which has taken the entire biomedical research community by storm, whether you do basic science or whether you're thinking about translation. And that's CRISPR-Cas and gene editing. And it's a great story because it comes out of one of the more obscure areas of basic science, studying yogurt and studying bacteria and their viruses. And out of this, we discover this system of CRISPR-Cas9 that is able to result in complex genomes being edited rather precisely with the enzyme plus a guide RNA to target you to the thing that you want to cut. Initially, much of this devoted to making a cut to knock something out, but increasingly, some of the newer versions of this being able to do an even more precise change of a single nucleotide. And I suspect in this room, probably more than half the people are engaged in one way or another with CRISPR-Cas experiments. Certainly, my lab is using this in many ways that we would not have thought possible and that have sped up the opportunity to do experiments, whether you're doing cell culture or animal models. So the big frontier now, I think, is to figure out how we take that set of advances and move it into the translational space. At least I think that's one of the big frontiers. Clinical advances are coming. We've already seen some pretty exciting preclinical studies. The mouse model of Duchenne muscular dystrophy by delivering Cas9 in vivo with an appropriate guide to turn the Duchenne muscular dystrophy in that mouse into something much milder, more like a Becker. That partial recovery of muscle function shown in that situation. Obviously, the issue here is delivery. Can you get the whole machinery into the cells that need it? And of course, we've been working on that for a long time with gene therapy, and we can take advantage of some of those things. Imagine that you had a series of vectors, maybe they would be AAV or maybe some other vector, that were precisely tuned to the tissue that you wanted to do this delivery. And imagine you had a whole lot of GMP scale Cas9 enzyme, and you could make your RNA guide also a GMP capability. Think of all the different genetic disorders that you might start to tackle in that way. And this has already been approached in an ex vivo way by having the cells taken out and treated, which makes it much more under control. And you probably know the first gene therapy protocol has now been approved by the RAC and will enroll its first patients in the next month or so. Chinese have already started this same protocol. This basically takes out T cells for people with cancer and disrupts their endogenous T cell receptors and the PD-1 gene in order to make those immune cells particularly ready to go after the cancer. It's a very clever effort that Carl June has put forward. And of course, because it is ex vivo, the mechanics are somewhat easier. I guess I'm getting more and more excited about the potential of how this could be used. And I want to tell you about an example that I think we could potentially see real progress in as little as 5 years, and that's sickle cell disease. I brought together just a month ago the individuals who are working in this space who are already doing bone marrow transplants for sickle cell disease, but most sicklers don't have a matched donor, and so it's out of the reach, and of course the risks are substantial because you're talking about a transplant that's going to require lifelong immunosuppression. Suppose you could use the patient's own cells, take them out, correct them, and put them back. Maybe that situation where the first molecular disease, which was sickle cell disease, would finally yield this kind of approach, because anybody who's watched progress here has to be frustrated about how we still don't have answers for a disease that we've known the basis of for a long time. Described in 1910, inheritance shown to be recessive by Jim Neal and separately by Linus Pauling in 49. Ingram showing the genetic basis in 57, for heaven's sake. hemoglobin genes cloned. This is what I did as a postdoc. I was working in this area, identifying variants in the genome that resulted in keeping your fetal hemoglobin turned on, so-called hereditary persistence of fetal hemoglobin, which turns out to be really good for you if you have sickle cell disease. Your fetal hemoglobin can save you. Hydroxyurea, yes, the first approved drug, not widely available as it should be and not as successful as we wish it was. And now bone marrow transplants, but few patients have a match. So why not contemplate here that we could tackle this, maybe as one of the first really big success stories of CRISPR-Cas treatment of a genetic disorder. Take out those hematopoietic stem cells from the bone marrow, utilize the culture systems that have been well developed, basically use CRISPR-Cas either in the most elegant way to fix the sickle mutation, but if that's too hard, you could actually create hereditary persistence of fetal hemoglobin quite easily with a well-targeted knockout, and I know about 4 ways to do that, and that would result also in a cure. If those cells are making enough fetal, the sickle doesn't matter so much. Expand those. Now you have to do something to make space in the marrow to put this back, and that's been a big deterrent because a full chemotherapy busulfan bone marrow eradication has a lot of risks, but there's some very interesting new developments that would make space in the marrow without producing quite the same level of risk and toxicity that can also be explored. I believe if we put together the right team, and that's something that Gary Gibbons, head of Heart, Lung, and Blood Institute, and I are trying to work on right now, involving both public and private sector experts, we might be able to put this plan in place, try it out, of course, initially on animals, including primates, and perhaps have clinical trials for humans of this CRISPR-Cas-mediated cure within the next few years. And I, for one, will be pushing as hard as I can to see that happen. This would be an incredible success story after many decades of frustration with the first molecular disease. Well, I want to move then to maybe a newer program, which is kind of basic, and it's kind of translational, and it's kind of all of that. In fact, it's called All of Us, and tell you a little bit briefly about this very bold initiative, which is at a critical space because its beta launch is about to happen maybe even next week, and you're going to be hearing a lot about this. The motivation for this is a realization that medical research opportunities have really evolved in interesting ways over the course of the last couple of decades. Patients are much more interested in being engaged in research, not passively but actively, but as partners. Electronic health records, clunky though they are, have come along and do have potential to be utilized in research, and many people are beginning to figure out how to take advantage of those in ways that we never could have done with pencil and paper records. Technologies advancing all the time. I don't know how many of you are wearing some sort of sensor. I have my Fitbit on today, and I left my Apple Watch at home. These kinds of things provide an opportunity for research to collect an awful lot of real-time data about what's happening to individuals as they go about their daily activities. And genomics, especially now with the increasingly low cost of doing not just genomic DNA sequencing of the germline, but also looking at gene expression in accessible tissues. All of those things sort of coming together. So what would you want to do? Well, you'd want to have a lot of data, and you'd have to make sense out of it, and you'd want a very large group of individuals, ideally who are quite diverse and represent our nation, and you'd want to follow them for a long period of time, collecting every possible kind of data, and you'd want to put all that data out in a public database where every researcher who has a good idea can use it and get access to it while still protecting the privacy and confidentiality of the participants. That's what we're trying to do with All of Us. This is a cornerstone of the Precision Medicine Initiative that Obama announced a couple years ago. It is a new model of research. It is based on a real collaboration between the partners who are going to be making accessible all of this information. And it is unbelievably large scale. 1 million or more volunteers is what we're aiming to achieve. Participants will be able to see results from their own data to the extent that they want information, they're going to get it. And it will have a rich data resource, including biospecimens, the Mayo Clinic serving as the biobank with capacity for 32 million biospecimens, and those are already starting to appear as we're testing out the systems. And again, the goal here is not just to study the treatment of chronic illness, although we'll get a lot of opportunity there, but also to study prevention, because many of the people involved in this, coming as a broad spectrum across ages and genders and geographic areas and ethnicity and socioeconomic status, will be, at the present time, reasonably healthy, and we can follow their status over time. The beta launch of this, testing out all the systems, will be started with our system leader in Pittsburgh. By the way, there are 60, count them, 60 grantees involved in this very large network. The national launch is aimed for this coming fall, and you all will be invited to take part in this, so you need to watch for that opportunity. If you want to know more about how to be sure you get notified, if you go to joinallofus.org and put in your email address, you'll be getting updates on the status of getting all of us off the ground. I recruited to run this, not an academic individual, but the guy who ran the health division of Intel, Eric Dishman, who is himself a cancer survivor and a real go-getter with more energy than any 10 people I know. And he has come in with great energy as the new face of this precision medicine initiative. And we've also recruited Dara Richardson-Heron, who's previously the medical director of the YWCA, as the chief engagement officer, to be sure that we are reaching out and connecting with participants in a unique and meaningful way, so that they are not human subjects. No, that will not be a term we ever use. They are partners, they're participants. They're all very much at the table. Let me just quickly show you a little image. I hope the audio will work here, of the kinds of thing that we want the public to see about this, as a brief video put together to try to explain it.
We are one nation, people. When called upon to give from within, we come together and find that our capacity to help others is limitless. Here, we are fearless. What lies inside all of us is more than data. It's life. It's more than insight and medical research. It's vision and honor and compassion. What's flowing through America's veins is its diversity. The next great breakthrough will be found in each and every one of us. And what we find there will unlock mysteries, heal the sick, and eradicate disease. We ask for 1 million individuals to come forward and stand on this landmark in history. We ask America to do, once again, what she has always done. Lead the way forward. We're one nation, one people, but all of us are different, and it's those very differences that will lead to answers for generations to come.
So watch for that, and hopefully some of you will find this an interesting thing, not just to watch, but to take part in. It will be an amazing national experiment. It goes back to Framingham, 1948, except now we're trying to do this at a scale that's 40 times larger, with a lot more kinds of data and covering all possible disorders that you could learn about with 1 million people. Certainly cardiovascular disease, but many other things as well. Well, let me finish by pointing out a few other research essentials, and particularly the most important research essential that we depend on for these dreams to come true, and that's the workforce. That's all of you. We are very interested in trying to be sure that we're inspiring the creativity and the confidence, particularly this next generation of scientists, that this is the greatest moment in history to be part of this engine of discovery called biomedical research. And we want to be sure we're providing, within the limits that we have been given budgetarily, those opportunities. With regard to the budget, let me just say that although there has been considerable anxiety, and I have shared that over the course of the last 14 or 15 years, since 2003, the budget for the NIH was gradually losing ground in terms of purchasing power and was hammered by other things like sequester. The last 2 years have turned a remarkable corner in the sense that in FY16, the Congress came up with an additional $2 billion for NIH. And if you noticed, just 2 weeks ago, they did it again for FY17, another $2 billion. strong bipartisan support in both houses of the Congress, and they are talking about being determined to make this kind of stable upward trajectory the pattern and not just the exception. And after doing it 2 years in a row, let's see what they can do for 3. They're certainly promising to try their hardest. So I'd want to reassure people who are perhaps a little worried about some of the issues and the noise out there that we do, in fact, have a case that's being made very effectively by all of you, by the work you're doing, and that people are in the Congress paying close attention. And they are very much on the side of trying to see how to nurture this enterprise, recognizing that it has been one of America's greatest success stories, both in terms of advances in health and advances in the economy. And that certainly is one of the reasons why I'm happy to be able to go down to the Hill, which I do at least once a week, and talk to members. And I can't remember a single one of those meetings, now with probably as many as 400 members of Congress, that went badly, because the story is so compelling and it tells itself. Congressman Pete Sessions, who invited me to be here today at SMU for a conference over there and to give the commencement address tomorrow morning, is one of those people who's very much on the side of supporting this enterprise. So we have lots of friends in this space across different party lines and other kinds of political views. Medical research continues to be one of those last areas that is truly bipartisan and has not been caught up in what otherwise is a pretty nasty time in the politics of Washington. But let me say what we're trying to do then with those resources. One of those is to, in a time where people are worried that maybe our peer review system discourages people from taking risks, we would not want that to be the case. So we have these high-risk, high-reward programs that support innovative researchers with potentially transformative goals, including some that allow you to skip the postdoc if you're ready for independence. An array of programs also to enhance diversity. This is a personal priority for me. We have not succeeded over several decades in making a career in biomedical research welcoming to people from all different varieties of backgrounds, and we need to fix that, and we're now investing about $50 million a year in new programs to try to achieve that and monitoring them very closely. Programs we've done in the past to try to achieve diversity all too often have not been evaluated in the sense that you could really tell what was working and what was not. And I'm tired of anecdotes. I want data, and I want to see what we can do here that really works. New programs to reverse the slide in physician scientists. That's crucial. If you look at the number of individuals who have MD degrees who are going into research careers that involve meaningful research contributions, which has got to be a significant fraction of your time, at least I would say 50%, that number is dropping down steadily. And we have some new ideas that are going to get rolled out in the course of the next few months about how to make that kind of career more appealing and more supportive. And policy changes also, not just to look at the early stage investigators, but once you get on that rising curve of a successful career in research, how do we make sure you sustain it? Because getting people started requires us also to pay attention to nurturing their careers. And we have policy changes, some of which are a little controversial, you might have heard about in the last week or so, to try to figure out how we can be the best stewards of the money that the government is able to distribute for research. These high-risk, high-reward efforts, I just want to mention the 4 categories because you have been very successful at this institution in applying and successfully getting those. The Early Independence Award, for instance, is this program that allows you to skip the postdoc. There's somebody who just got one of those awards. The New Innovator Award. You can't apply to this unless you've never had an NIH grant and you have a bold idea that it would really be groundbreaking, and we don't expect you to have a lot of preliminary data. You've been successful in that space too. The Transformative Research Awards oftentimes are team efforts. They also have to be really bold and don't require you to write this long, complicated set of preliminary data. And you've been good at that too. And one of the very first Pioneer Awards back in 2004 when we started this is Steve McKnight, who's obviously been incredibly creative all the way along. So if you're interested in those kinds of programs, you can read lots more about them on the NIH website. And if you're interested in trying to understand how it is that the NIH, given as we are by the public, $34 billion a year, how do we try to figure out how to use that in the most effective stewardship fashion? This document, which is only a year old, is actually pretty readable. Most strategic plans aren't. This one will run you Yeah, if you spent 45 minutes, you could pretty much get through it. There's a lot of examples here of things that I think demonstrate what we're trying to do. And it goes through this virtuous circle between fundamental science, health promotion, disease prevention, treatments and cures, and how we try to set priorities, how the peer review system works, and what we envision as some of the areas of greatest opportunity might be worth a look. So I have already used probably more time than I should have, and I want to have a chance to engage in conversations with Dr. Russell. I'll just finish with this quote, which I think applies particularly well in a place like this that has been so much devoted to trying to take science and get answers to medical. And this, of course, comes from the father of internal medicine, Sir William Osler. And this comment just totally sums up what we are trying to do today, to wrest from nature the secrets which have perplexed philosophers in all ages. Boy, we're trying to do that. Talk about the brain, for instance. To track to their sources the causes of disease, yeah, that's the applied part. To correlate the vast stores of knowledge, that sounds like big data to me, that they may be quickly available for the prevention and cure of disease. These are our ambitions, and I bet they are yours also. Thank you very much.
Appropriate talk show format here.
Yes.
So we're going to do this in two ways. Because of the size of the audience and the limit on Francis's time, I have a couple of questions that you all submitted and we collated, and then we'll also take a couple of questions from the floor. So I think I'll start and ask you a question.
And it's wonderful to be on your show, Oprah.
Oh, thank you. Yes. So I'll leave that one alone. You had an editorial, Francis, recently in the New England Journal of Medicine about the 21st Century Cures Act.
Yes.
And I wonder if you could comment on how you think that will reduce the regulatory burden of doing experiments on humans and animals.
So 21st Century Cures Act was the most significant piece of legislation affecting medical research in about 10 years since the last NIH reauthorization. And it took place in an evolving way to have many components over a 2-year period, and many, many shout-outs are due to Fred Upton and others in the Congress who tirelessly stuck with a program that many people thought multiple times was not going to make it over the finish line, and got this bill passed by dramatic bipartisan support during the lame duck period back in December. We, of course, are thrilled about it because of what it has provided in terms of funds for research. So there's serious money in there, about $4.6 billion total over a period of 10 years for the Brain Initiative, for the Precision Medicine Initiative, for regenerative medicine, and for the Cancer Moonshot. And those funds are separate from the regular appropriation, so that means that they can, in fact, be reasonably well counted on over the coming years, and we can plan on the basis of that. But there are also a lot of policy issues in that particular legislation, many of them focused on FDA, but some of them also on NIH. There were some things in there we loved. They now allow scientists who work at the NIH to actually go to scientific meetings without a whole lot of barriers put into place, which had been an issue. They got rid of a piece of previous legislation that has the most ironic name in history, the Paperwork Reduction Act, which for us was the largest producer of paperwork that you can imagine. We were freed up from that as far as scientific research. But yes, there was also an effort here to try to address what is seen as a serious set of burdens that institutions have to deal with in terms of requirements that the government places upon you for the privilege of getting a grant from the NIH. And we know that that adds to a lot of hours and a lot of people, and that some of this is actually not really very defensible. I'll just focus on one that I'm hoping we can, in fact, get fixed even before this policy board that the 21st Century Cures Act suggests should be formed gets in place, and that's effort reporting. Do I hear sort of an amen about effort reporting as a useless thing that we all are required to do? And we've actually done pilot projects to show that there's a simple way to do this that is vastly less trouble and gives the data that any reasonable person would want to see. But we've not been able to get it across the finish line because of resistance from some of the auditors and the inspectors general. I think we could fix that. But that's just one of several. I'd like to engage right now in a series of conversations with our grantee institutions about some of those barriers. Are we making the conflict of interest regulations as important as they are for our credibility, but are the way we're implementing it unnecessarily complicated and bureaucratic? We're ready to have that conversation.
Mm-hmm. Fantastic. I'll switch to taking a question from the audience. We've got Dr. Sandy Schmidt, our chairman of cell biology, so be prepared. This is going to be a tough one.
Thanks very much for coming, and thanks also for your concern about young scientists and mid-career scientists. You mentioned some controversies, so I wanted to ask a two-part question regarding the 21-point system that you're rolling out. So the first is, when modular budgets were implemented in the 80s, one module covered a postdoc and supplies, and today it takes 2 modules. So I wonder if you were considering increasing modular budgets and encouraging justification of non-modular budgets to encourage ambitious research projects. And second, are you concerned that this 21-point system might limit collaborative efforts across laboratories, and is NIH taking any steps to encourage effective collaborations?
Yeah, thanks. I appreciate the question, and we're in the midst of a very intense conversation about this proposed change in the way in which we review investigators who have already a very significant level of grant support. And this is not a place where decisions have been made. I want to reassure you, every one of the NIH advisory councils has been engaged in this conversation. This is council meeting time, it's May. And my advisory committee to the director will discuss this in a major way on June 8th, and that will be publicly video cast if people want to watch that. They're already working hard at it. We don't want to have this have unintended consequences. But I think anybody who hasn't looked at the data might want to do so, and it's something you can read if you go to Mike Lauer's blog, which is called Open Mic, oddly enough, and you can see what we have learned now that we have better analytics. And maybe this fits with what your intuition has been, but now it's not intuition, it's data. That if you look at productivity as measured by not a crummy metric of productivity, but the best we've got that correlates really well with expert opinion, and you look at what happens for an investigator who has 1 grant, and let's just talk about R01s, or 2, or 3, or 4, the more grant support that investigator has, the more productivity they have, but the sort of productivity per buck starts to flatten out. When you get about 3 R01s, it's clear, and this is human nature, that investigators are perhaps not able, with their limited bandwidth, to continue to have the full attention of everything that they're doing. And if you look at that, now admittedly, this is a small proportion, maybe only 3 or 4% of NIH-funded investigators are in that group that have 4 or more R01s. But it does look as if that may not be the best way for us to get the maximum out of the dollars we have. When you look at that, and when you look at that, our demographics are such that early-stage investigators and mid-stage investigators are having a tough time right now, and they generally don't have 4 or 5 or 6 R01s. Maybe this is an opportunity to spend the money in a better way. And this is not sociology or social activity. This is based on the data of what we can see about productivity. So the proposal, and it's been modified quite a bit since the initial rollout, and we might just focus on RPGs and not include things like cancer centers and training grants and so on, would be that we would have this threshold where if somebody went above, effectively, 3 R01s, that would be looked at very hard as, is this the best use of the money? And we would even discourage people from continuing to apply for more and more R01s and let that money instead go in another direction. That's the proposal. It's very open for discussion. The modular grants are a significant part of that. I want to mention one other thing, and that is this new approach which the General Medical Institute is implying now pretty much to their whole portfolio, is let's get into a different place where investigators have the opportunity to apply for 1 grant that covers what they're doing, the R35 as it's called, It doesn't require a vast amount of preliminary data. It doesn't require you even to stick to what you said you were going to do over a 5-year period if you come up with some better approach. It's more like being a Hughes investigator or an intramural scientist at NIH, where we trust you to have a good idea if you pass that review. And then you get that sense of support, and you don't have to spend all your time writing another grant and writing another grant, writing another grant. You can actually do the science. And I would encourage all of you, as you look at this, many of the institutes are now providing these R35 opportunities. That might be a better way to go for a lot of things that people want to do. And that would certainly get you out of worrying about this 21-point score.
If I could just extend that a little bit and ask you a related question, is there any evidence in that data that generated bang for the buck, or the analyses there that inform as to which style of grant provides or produces the most high impact research? Is it an R01? Is it an R21? Is it
It's all in there, and we're going to make all that data available so people can actually do their own analyses. It's interesting that one that we seem to get the most out of is actually these HR awards, the high risk, high reward awards. The number one sort of producer of exciting data that we have data on is the Pioneer Awards, where you get 5 years worth of support, and they're intended to be highly innovative. There's another question behind there. I don't know if you can see.
Andrew? Andrew Zinn, our Dean of the Graduate School. And
yeah,
also
Dr. Collins, you presented some reassuring numbers about the NIH budget, or at least the budget request, but there's still an unprecedented feeling of threat among the scientific community, to wit, the March for Science. And I wonder if you would comment on what you feel is the most effective way for the scientists and scientists in training to advocate for the value of research to the public and the politicians.
So I think the most effective way is to get to know your elected representatives, invite them to come to your institution. You all are in a state that has a lot of very significant leaders who are steering the ship on Capitol Hill. And to the extent that you get close to them, as I know you have been with Congressman Sessions, because he's told me many times about how much he loves to come here, that is the best thing you can do. Those are the way in which members of Congress really begin to get it about what is medical research all about. And oh my gosh, it's going on in my state or my district. It's not all happening there in a bunch of buildings in Bethesda, Maryland. There's still a lot of confusion about that. So yes, invite them, call them, go and see them when you're there. Tell them what you're doing. Bring along a postdoc who's really excited about a new finding, or a graduate student. Bring along a patient so that it becomes personal. And don't be shy to tell stories. Congress loves stories. Statistics are fine, but stories are better. Stories of discovery, stories of medical advances. Tell them stories.
Thank you.
Okay. I mentioned that we had people submit, from the faculty, submit questions, and I think your mom was amongst those who submitted questions. So you've been director, as Dan pointed out, of the NIH for 8 years now, or almost 8 years, and you've accomplished a lot from the Pioneer Awards and others. Which of your accomplishments are you most proud of?
Oh, gosh. You know, it has been an incredible privilege to be able to stand at the helm of this ship. And I, by the way, did not expect that when I came here I would still be the NIH director, so what do you know?
True survivor, maybe. Yeah.
I sent in my resignation letter as required as a presidential appointee, and figured that on January 20th at noon I would start doing something else. But on January 18th, I got an email from the White House saying, your resignation letter has been rejected. They didn't explain the reason. Maybe it was the grammar, I don't know.
That's too long, maybe.
But it was. But it's just the same, incredible privilege to continue to be able to play this role and work with the amazing team of institute directors, all of whom are world-class scientists and leaders in their own right. In terms of accomplishments, it's all been this sort of team effort. I look at organizationally, the one thing early on that I got to do was starting the National Center for Advancing Translational Sciences, NCATS, trying to be sure that we were filling in some of those valley of death problems that kept basic science from finding its way to the clinic. And over the last several years, these large, organized but very much built from the ground up projects. The Brain Initiative, the Precision Medicine Initiative, the Cancer Moonshot have been, I think, very gratifying. And the other thing that I've had a privilege of doing is working more closely with other government agencies, the FDA, the CDC, and CMS. We have close senior leadership connections with them to get things done. Likewise with the Gates Foundation, working in global health in a partnership that's much closer between NIH and Gates than has happened before. All of those things make this pretty amazing as an opportunity.
Yeah, well, we're certainly grateful for your leadership. I think we have time for one more question. Mark Diamond, a physician scientist and neurologist.
Yes, thanks for coming. In 1957, I believe it was 57, they launched Sputnik, and it triggered a huge investment in science and technology and enthusiasm across the population to go into these areas.
Yeah.
And given all the immense potential right now for scientific discovery, do you have any ideas about how we can create another Sputnik moment that can get a lot of our smartest young people to want to come into biomedical research?
That's a great question, because that clearly was transformative. I got interested in science because of a chemistry curriculum that was developed as a direct consequence of Sputnik and was extremely well designed and made you think about how to do science and not just read about it. So maybe I'm also one of the many people who is influenced by that. I'm not sure we can arrange something quite like that again. I do think there are some areas that we could perhaps do a better job of explaining why science is the answer. I mean, recently one can point to pandemics of outbreaks of disease, and certainly Ebola in that circumstance caught the imagination in a scary way of this nation, and certainly pointed to the need for medical research to be strengthened and move rapidly to come up with solutions. I think part of our issue is that our science education in K through 12 over the course of the last 20, 25 years has not kept up with the opportunities, and we continue to miss the chance to get young people excited about science. When I talk to scientists, I don't know, David, what your experiences was. Mine was that 10th grade chemistry class that said, I want to do this. I didn't know that until I went to that class, and I hear that from lots of others. And I worry about how many 10th grade classes now don't have that spark, and they basically are telling you to memorize stuff instead of how to use principles. So if you really wanted to try to turn this around and we can't have another Sputnik, we should really figure out how to reinvigorate our K through 12 STEM education. That's so critical, and we talk about it a lot, and there are a lot of people who care about it, but the trajectory right now doesn't look as encouraging as one would like. We have to get this fixed.
Okay, thank you. I believe our time is up. Let's thank Dr. Collins one more time.
So, Dr. Collins, I want to again thank you for sharing your insights and your time with us today at UT Southwestern. We'd like you to carry with it the UT Southwestern seal. So, it's a piece of that.
I know, I posted that.